Now there are 5 types of antibodies. So the first one that I consider to be kind of a boring one is IgD, immunoglobulin D, which serves as the B cell receptor. Next one, not very pleasant either, is IgE. This is an antibody that mediates allergic responses. Next we have IgA. It's a dimer, so it consists of 2 individual molecules, 2 individual antibody molecules linked together. And that's the antibody that can be found in the mucus, sorry for misspelling, in the mucous membranes or breast milk. It's basically first line of defense, breast milk or mucous membranes. Then we've got IgM. That's a pentameric antibody, meaning that it consists of 5 individual molecules linked together. It's a first response antibody. It's produced first in the process of infection. And what's important, that antibody, IgM, can activate complement. Finally, that last 5th type of antibodies is IgG. So IgG is the most abundant type of antibody. It can cross placenta, so it will provide protection to a newborn child, and it very actively activates complement. So let's talk about B cells first. And I want to quickly describe how B cells get activated. So imagine we have 2 different types of B cells. One type of B cell carries on its surface a certain kind of a B cell receptor. And here's the different kind of B cell. It carries on its surface a different kind of a B cell receptor. And all of a sudden, you have an antigen that looks like this. So this antigen will bind to its unique receptor on the surface of a B cell. So this population of B cells will be selected. This is called a clonal selection. The population of B cells that carries the same B cell receptor is called a B cell clone. So during the clonal selection, that's clonal selection, this population is selected to respond to that particular antigen. To the clonal selection, these B cells start to proliferate, meaning that they multiply, and differentiate, so they become specialized. This is called clonal expansion. So now you have a bunch of B cells with the same receptor, and that's going to be few populations. So you're going to have a population of a memory B cells and the plasma cells. And these plasma cells will start producing antibody molecules that match the antigen at their specificity. So this process of proliferation and differentiation is called clonal expansion. And once clonal expansion is done, you have a lot of memory cells, you have a lot of plasma cells. So we go to this memory plasma cells. Plasma cells producing antibodies.